In this video, you will learn about the role of calcitonin, parathyroid hormone, and calcitriol in the maintenance of blood calcium homeostasis. Maintaining calcium homeostasis is critical for normal physiological function. Calcium ions are essential for processes such as muscle contraction, neurotransmitter release, blood clotting, and intracellular signaling. Calcitonin is a hormone that decreases blood calcium levels. It is produced by parafollicular cells, also known as C cells, of the thyroid gland. The release of calcitonin is stimulated by high blood calcium levels, a condition known as hypercalcemia. Calcitonin indirectly stimulates osteoblasts and directly inhibits osteoclasts. Calcitonin is a hydrophilic hormone that binds to G-protein-coupled receptors located on osteoclasts. These receptors stimulate adenylyl cyclase, leading to the production of cyclic AMP and activation of protein kinase A. Within the osteoclast cells, this signaling pathway ultimately has two main effects. The calcitonin receptor signaling mechanism directly inhibits osteoclast activity to decrease calcium resorption from bone. Calcitonin receptor signaling is also responsible for indirect regulation of osteoblast activity. Intercellular signaling molecules produced by osteoclasts regulate osteoblasts to stimulate calcium deposition. As osteoclast activity decreases and osteoblast activity increases, calcium moves from the bloodstream into the bone. Parathyroid hormone, commonly abbreviated as PTH, has the opposite effect. It functions to increase blood calcium levels. PTH is produced by the chief cells of the parathyroid glands. The release of PTH is stimulated by low blood calcium levels, a condition known as hypocalcemia. Like calcitonin, PTH is also a hydrophilic hormone. However, PTH binds to receptors on osteoblasts rather than osteoclasts. Activation of these receptors stimulates the cyclic AMP and protein kinase A signaling pathway, leading to two major effects in osteoblasts. PTH directly decreases calcium deposition and stimulates osteoblasts to produce intercellular signaling molecules that stimulate osteoclast activity to increase bone resorption. Breakdown of hydroxyapatite releases calcium and phosphate into the bloodstream. In the kidneys, PTH increases calcium reabsorption. This reduces calcium loss in the urine. PTH also decreases phosphate reabsorption, increasing urinary phosphate excretion. Additionally, PTH stimulates the production of calcitriol. Vitamin D activation occurs through two sequential hydroxylation steps. The first hydroxylation occurs in the liver, producing calcidiol. In the kidneys, PTH stimulates the second hydroxylation step, converting vitamin D to the active hormone calcitriol. Unlike calcitonin and PTH, calcitriol is a steroid hormone. Because it is lipid-soluble, calcitriol diffuses through the plasma membrane and binds to intracellular receptors. In the intestines, the vitamin D receptor stimulates the transcription of genes involved in calcium absorption. This includes genes for calcium ion channels and calcium transport proteins. As a result, calcium absorption from food increases. Together, these mechanisms increase blood calcium levels and restore calcium homeostasis. Now that we understand how calcitonin, parathyroid hormone, and vitamin D regulate calcium homeostasis, we can consider what happens when calcium intake is inadequate. Dietary calcium deficiency remains common worldwide, and this presents a significant challenge to the maintenance of blood calcium homeostasis. Even when blood calcium levels remain within the normal range, important physiological adaptations are occurring. In many parts of the world, vitamin D deficiency is more common than inadequate dietary calcium intake. Vitamin D deficiency reduces calcitriol production, leading to decreased absorption of dietary calcium. As calcium levels decline, the body compensates by increasing PTH secretion. This increases bone resorption, leading to increased risk of osteoporosis. In children, vitamin D and calcium deficiency can result in rickets. Bones become soft and weakened, leading to skeletal deformities such as bowed legs. Milk is often fortified as an effort to prevent vitamin D deficiency. While milk fortification has significantly reduced the prevalence of rickets, vitamin D deficiency is still a major public health concern. Secondary hyperparathyroidism results from chronic hypocalcemia, commonly due to vitamin D deficiency or low calcium intake. Primary hyperparathyroidism results from an abnormal growth of overactive chief cells in the parathyroid glands, leading to elevated production of PTH. In this condition, elevated PTH causes hypercalcemia. Clinical manifestations may include bone pain, osteoporosis, kidney stones, abdominal discomfort, and calcification in the brain, leading to impaired cognitive functions. In summary, calcitonin, parathyroid hormone, and calcitriol work to maintain blood calcium homeostasis. While calcitonin lowers blood calcium levels, parathyroid hormone and calcitriol work to increase blood calcium levels. When these regulatory mechanisms are disrupted, significant skeletal and metabolic disorders can develop.